few lectures. Anything, something burning in your mind? You need that answer now? No? Anything? You went over the slides, right? I think you did, right? Okay. Even if you did not, I will accept your answer. All right. We're going to talk about the last two parts, which is control and prevention, right? Control and prevention of biofilms. So we learned so far how biofilms are made, what they do in terms of disease, infection, then problem associated with food spoilage, beneficial aspect, right? All kinds of stuff we learned. And now let's say at least control and prevention. When you talk about control and prevention, that means we are going to talk about mostly where we have undesirable property, where biofilm can cause problem, whether it's a product development, product safety, or shelf life, and also from the disease standpoint, if these organisms or biofilm can be eradicated or eliminated, and from the standpoint of therapeutic or clinical application, we have to know how to prevent or control, right? So I'll give you some bigger, again, like big pictures, some detailed information, but then you can read up a little bit more how specifically some of those things are done. Okay, again, same picture you've seen again, just to remind you what is, how they form, and what are the final steps, and of course, what are the different factors. Again, when you try to control biofilm formation or also prevent biofilm formation, you have to take into account how do they are formed, right? Why they attach, how they attach, right? All of those factors, and what do they do during biofilm formation, right? Again, all of those things we talked. Now you need to apply it, and that knowledge is going to be extremely important for our strategy. Exopolysaccharides, right? Polysaccharide, exopolymer, polysaccharides, proteins, eDNA, lipids, they are the major matrix molecule that hold these molecules together. Then, of course, the quorum sensing. So, if you know what makes these biofilms work, then these are the certain approaches you can undertake to help prevent or control biofilm formation. Okay. Throughout this lecture today, we're going to talk about individual steps, right? So if you just broadly take a look at it, say, can we use antibiotics or adjuvant agents or any kind of antimicrobial agents to kill microorganisms within the biofilm, right? Then quorum sensing inhibitors. We talked about quorum sensing, right? Can we use certain molecules which are going to stop the communication between microbes? So if they cannot communicate, therefore they cannot form biofilm, or they can continue to establish that biofilm-like structure where you can easily eliminate them. What about enzyme? Right? Can we use enzyme to break down the biofilm? Can we? Answer would be yes, right? Because the molecules we mentioned, polysaccharide, protein, lipid. If we have enzyme for those target molecules, you can easily disperse them, right? You can destroy biofilm. There are a lot of natural and synthetic antimicrobial peptides. We'll try to use some of those examples. Antimicrobial, peptide, or plant-derived polyphenol or essential oil, some of those are antimicrobial, right? They can also inactivate microorganisms. You heard about CRISPR-Cas, right? CRISPR-Cas9 is a gene editing tool, right? So CRISPR-Cas, it finds the gene target, and then enzyme Cas9 that can chop that, cut that section. And a lot of times that can be manipulated in a way you can control microbial growth or biofilm growth. I'm not going to spend any time talking about it. These are new technology people trying to use to control or prevent biofilm. Nanotechnology. Can we use nanotechnology approaches to control or prevent biofilm? I will show you some data where we used and other people also have used can be applicable for controlling or preventing biofilm formation. Okay. Probiotics, again, we talked about it. Probiotics can compete with microorganisms. They can also help reduce biofilm formation. Even though they themselves can form biofilm, but they can still help remove or prevent biofilm formation. What about this one? Biophysical chemical therapies. There are some fascinating research in progress. If you think about wound, right? Wound surface. And wound have typical biofilm, right? We discussed about that. And there are also nanotechnology based approach where you can on demand deliver antimicrobials. So that means your sensor can sense the environment. Okay? And then strategically you can ask your sensor or device to deliver antimicrobial agent. One of the applications is the ozone. You heard about ozone, oxygen, O3, right? Ozone, and it's highly antimicrobial. And ozone, of course, we have the ozone layer on the top, right? We are depleting ozone layer, and that's the reason we are getting a lot of sun ray coming through. And that's part of the warming trend we are seeing. So we have to protect our ozone layer on the environment. But here in the ozone, we use ozone as antimicrobial or sanitizing agent. And so some of this wound application, I don't have any data or paper to show you, but that's why I'm trying to indicate that if you have an ozone molecule and you can strategically deliver ozone onto the wound surface or on the biofilm surface, you can control that. And I thought these are some of this fascinating ongoing research we are doing, or some of the people in engineering, they are doing this. If you got general idea, because I'm not going to present any data on this, but just trying to tell you these are the new generation of research in, we call it smart sensor or smart technology. It's on demand, will deliver bioactive molecule so that you can control the microbial growth, okay, on surface. And these are specifically designed for a lot of this biofilm in the wound surface, on the skin damage, okay, external. So we're going to talk about individual items briefly. So disinfectants, right? I mentioned that chemical agents, enzymes, probiotics, bacteriocins, bacteriophages. Bacteriophages, I didn't mention that in the graphics. Nanoparticles, QS inhibitors, and also called micro nano bubble. Everybody heard about micro bubble? Heard about nano bubble? Anybody? You did. The micro bubble or nano bubble, I'll show you the schematic. It's literally the bubble. The water bubble forms, right? And then when the bubble bursts, it has a certain energy that can kill microorganisms. It's fascinating, right? And if you have a nanostructure of that bubble, it can penetrate in certain places in the microbial community, and then it can also kill them. So it's a new generation of processing technologies people are using it. So I will give you an example. Ultrasound, photoradiation, okay? All of these are done. So food industry, so disinfectants kill or inhibit microorganisms that can spoil food. Can be indirect or incidental food additives used on food contact surfaces. Applications are regulated by EPA, so you cannot use disinfectants randomly. Usually they are regulated. And some of them are used on raw meat or poultry, especially for washing, surface washing. We'll use a lot of like a low doses of chlorine, for example, right? Or ozone, I mentioned. And they can be used, or acid, okay, can be used to control microbial growth on surfaces, right? If they are on the meat surface, can you use this washing to remove the biofilm, okay? And so these has to be food grade, because we are all going to consume that afterwards, right? And of course, we wash them to remove residual disinfectants, but still, If there is any residual left, that could be a problem. So these are the molecules, the treatments I mentioned: sulfide, sulfur dioxide, chlorine, ozone, quaternary ammonium compound, that's called quat, peroxides, like hydrogen peroxide, they use that. And these are the chemicals or disinfectants are used on surfaces to inactivate microorganisms. Factors that may affect surface disinfection. So we are trying to kill microorganisms, right? So if there is a biofilm form, of course, that may affect depending on the level of biofilm formation. What type of substrate you have, the type of microorganisms you have, or what are the conditions, external conditions. So all of these factors, I didn't mention that, you can look at it. If it is a biofilm, you want to know how old is this biofilm, right? If it is a newly formed biofilm, it's easy to remove, right? If it is a very old biofilm, I showed you some of the pictures, it's very difficult to remove them, right? So the age of biofilm is going to be problematic for how effectively your disinfectant can work. Everybody following me? So these are 4 factors. Within the factors, you can see, read it carefully, how much EPS molecules produced. Is the quorum sensing active? Persister cell. If you have a persister cells, they may not uptake any of this disinfectant or antimicrobial. I know you cannot read this. Can you read this from the back? Very rarely, right? Or unless you have a very good eyesight. I know I kind of feel bad that when the slides are smaller, I never like that, okay? But these actually came from a paper, so I couldn't do anything. I didn't want to manipulate it. But just trying to give you an idea, what are those factors affect disinfectants? Disinfectant may not work. So pay attention to this. Degradation of degradative enzymes, neutralizing chemicals, microenvironments, horizontal gene transfer. So everything about, we talked about biofilm characteristics. All of those factors can affect the efficacy of disinfectants. Okay. Substrate, we're talking about substratum, and it's the surface, surface charge, surface topography, weightability, conditioning film, sediment accumulation. So where the biofilm is formed, right, you need to know the surface chemistry, surface properties. All of this is going to affect if the disinfectant is going to work. And this is extremely important, because if you are assigned to take care of safety of food or molecules or drugs, anything produced in that environment, if there is biofilm forming, they're going to ask you, how do you get rid of this biofilm? And then you are looking into all of these factors. As an expert, you're going to tell that, well, some of these conditions you are using. So whether you are using stainless steel table versus wooden table. Okay. So if you are preparing food, it's a practical application. I'm trying to use it. Which surface is most desirable for application of disinfectant? And what is that surface we normally use on the food industry or pharmaceutical industry or anywhere you do? The steel is the best preferable, right? You can easily sanitize them, and their surface generally discourage formation of biofilm, right? The way the structure is. But if you look at the wood or plant-based or any other surface, that may be favorable for microbial colonization, right, and biofilm formation. And if that happens, then you cannot wash them readily, right? And you cannot clean them properly, and then the wood would be reacting or responding to the chemicals or sanitizers you are using, right? So the surface topography, it has to be smooth, right? If it is rough, then chemical may not reach in every site. Okay. I'll give you an example. And even if you have a scratch in the nanoscale level or microscale level, that still can prevent your disinfectant from getting into that area. So that is the harborage site where microbes stay and grow. And this is so fascinating. You don't feel it, but that's the true application. A lot of people spend their entire life trying to figure out all of the surface chemistry, surface topography. how that can help the biofilm to persist for extended period of time. So that become a continuous source of infection or infective agents if you are producing any food or if you are producing any pharmaceutical drugs or molecules or a hospital setting, clinical setting, where is this pathogens coming from? Okay, and what this is saying is that this is the only tool you have, actual powerful tool in your hand to get rid of a lot of those biofilm or biofilm cells. Everybody clear on this? Okay. So when I mentioned that the substrate is going to be important, then also the bacteria, what type of microbes you have. What did we learn? Even if you look at Staph aureus, right? And if you take a multiple different strains of Staph aureus, and you already know that the biofilm characteristics may depend on the strains, right? Sometimes maybe serotype. Not, I also showed you data for Listeria, right? And Listeria, you can see some of them form strong biofilm, some of them form weak biofilm, right? So within the same species, you may find variation, right? And you need to know which organism you are trying to get rid of. Is it Pseudomonas versus E. coli versus Salmonella? All of those different organisms has different criteria for forming biofilm. Some form strong, some form weak. By now, you all know that, right? And you need to take into account when you are trying to apply this disinfectant. Then also surface charge of the molecule, right? Or the bacteria or the microbes. What is the surface charge of bacteria in general? Is it positively charged? Is it negatively charged? Charge-wise, what's your knowledge? Negatively charged, right? Okay, those who do not know, bacterial surfaces are negatively charged. Okay, so if they are negatively charged, wherever you have a positive charged surface, they are going to form a strong biofilm. But if it is a negatively charged surface, then you know it's going to repel, right? And so can you create environment or surfaces that you have that type of charge distribution? Now you can prevent biofilm formation, whether it's in the catheter or medical devices or on any surfaces. Okay, so that's why I'm saying today's lecture should be the most important one. You learn everything, how they form, what are the foundation, and now we're trying to get rid of them. We don't want them. What can we do to prevent it? Shape and size also important. Appendages. Appendages means what? What is our appendages mean? You all know that, right? Appendage, that terminology, English. Appendages means arms and legs, right? The bacteria has arms and legs, right? Come on, we talked about, right? Flagella, right? Fimbriae, pili, all of those are called appendages, right? So this terminology they use here, they're saying that appendages. So some are expressing fimbriae, they will be more attaching, right? If they're producing flagella, they will be going and attaching themselves, right? So all of those factors contribute to the type of biofilm we are going to see. Motility, microbial metabolites, tolerance and resistance mechanism to antimicrobials, right? All of those factors are going to contribute how strongly biofilms they are going to form and whether this disinfectant is going to work. The last part is the external conditions. We all know that, right? Temperature, pH, humidity, nutrient load, ionic strength, hydrodynamic conditions, antimicrobial agents. So these are some of the external factors, right? If the pH is low, a lot of times they may not colonize, right? If it's high, neutral, oh, well, perfect condition. They can grow, multiply. Same with the temperature, right? Today is cool, but 2 days ago it was pretty hot. So if you have a surface or surface exposure to microorganisms, so they would be growing much faster, right? When high temperature. And so they could form biofilm quickly and rapidly. So if you have a dense formation of biofilm, the disinfectant may not work well that day. Or also the function of disinfectants or chemical sanitizers, a lot of those are temperature dependent. Did you know that? So what temperature generally do we use to apply disinfectant? Just a common sense. So if I give you cold water, ice water, right? Or I give you warm water, which one is going to work best? Common sense. Warm water, right? Because that's going to dissolve all the chemicals properly, right? And it has a greater access to the microbes. Again, these are all common sense. So if you put sanitizers at low temperature, it's not going to dissolve, it's not going to diffuse well, it's not going to make a good contact, right? Because that's a low temperature. Your surface properties of the microbes also going to change. They're going to behave differently. Are you all with me so far? Okay. So all of those factors play a role how well your disinfectant or any antimicrobial is going to work. So I spent time on this kind of setting up the foundation for you to understand. Okay. Now let's talk about enzymes. I think all of you, if I ask this question, even if I don't go with this slide, you should be able to answer it. We already established what are the metrics for biofilm formation, right? I just mentioned that. You have polysaccharide, you have protein, you have lipid, you have DNA. So name one enzyme for each matrix. Polysaccharide. You got a lot of different enzymes, right? Carbohydrate breaking enzymes. Why not use those enzymes to break down the carbohydrate? Protein, protease, right? You got varieties of proteases. Use something broad spectrum protease rather than narrow spectrum protease, right? So you can break down the protein. What about lipid? Lipase, right? So why not use lipase to break down the matrix? Then DNA. Easy, right? DNase. Break down the DNA. So that's how they are trying to apply enzyme to break down the matrix. Therefore, the dispersion happens. Well, these enzymes may not be able to kill the microbe, right? They cannot. Generally, they don't, or they may not, right? Unless these enzymes are integral part of the structure. So obviously, you have to use something else. So a lot of times we use enzymes together with disinfectants or antimicrobials, so that the drug penetration would be much easier and faster to each of those organisms. Again, all of these are common sense. I want you to use common sense. Don't think too critically. So maybe there is a more important in-depth or deeper questions to answer. But if you look at this, proteases, cellulases, polysaccharide depolymerases, alginate lyase, these are all of those conditions you can use to break it down. So if you look at these polysaccharide breaking enzymes, they have listed few of them. Okay. Again, go over this slide. The DNA, then protein. I mentioned this. Then also they used acylase. We'll talk about that. That's going to break down the quorum sensing molecule. Again, that's the enzyme, right? Then lipase, and then also cellobiose dehydrogenase. So any other molecules you have, it can break it down. That works, right? Okay. Antimicrobials. So I may go over the bacteriocin a little bit. You already have general idea about bacteriocins. Many of you, right? You heard about it. So bacteriocins are small molecules or peptides. These are always protein or peptide, and they are going to be released by certain organisms. And usually the history behind bacteriocins is that when microbes growing in a community or in an environment, they want to compete, right? For the competition purposes, they will secrete some small molecules, and then they can kill their competitor, right? So if you have an environment, they are all growing in this community, and say, another definition you have to remember is that they are effective against closely related organisms. You heard about that? Closely related organism. What does it mean? If I'm a gram positive, I will work against other gram positive in that environment. Okay? And if it is closely related, then it may not work against gram negative. And so what they're trying to do is that in a given environment, all gram positive organism wants to grow. And so to outcompete them, some of them would produce this bacteriocin historically. So they get an advantage in that environment. They will grow, they will utilize nutrient, they get an upper hand. So it's killing other gram positive organisms. Very rarely we will find broad spectrum except the reuterine. Somebody mentioned that yesterday. Other than that, actually they are very narrow spectrum. You can make some bacteriocin against other organism work is because their target is cell wall, okay, I'll tell you a little bit later, or the cell membrane. And a lot of times in gram-negative organism, bacteriocin may not reach to the side. So sometimes if you break down the outer layer, this is what we are discussing, circulating agent or hydrostatic pressure or injury, that time bacteriocin can get into the unrelated organism, can kill that cell. So you have to do some special treatment to be effective. What they do pore form, inhibit protein synthesis, also degrade RNA. Okay, that's the general function of the bacteriocin. And there are a lot of review articles, a lot of people worked on it. Early in my professional life as a graduate student, I worked on this molecule, pidiosin ACH. So you can see class 1, class 2, and class 2 again subdivided, and then class 3. It's based on the molecular weight, how many different types of amino acids you have, whether it's a hydrophobic, sulfur-containing amino acids, disulfide bridge. This is one of the characteristics, and these are the different names and different organisms. Again, if you are interested, you can study a lot of those. A lot of work has been done, and this has been used in terms of research wise. A lot of data available, a lot of publications available, but niacin is the only one is approved by the FDA, Food and Drug Administration. And I don't know why other bacteriocins are not approved, but nonetheless, this is the only one been approved. And if you look at the structure, it got some modified amino acids. Definitely that contribute to the structure. Anyway, so how does it work? As I mentioned that many of them are actually pore formers. And if you look at the class 1, if niacin, what they will do, they will bind, go through the peptidoglycan layer, and then interact with the cell membrane, and the way it interacts, and then ultimately form a pore. So the pore formation basically very similar to the class 1 and class 2. You can see that integrate and then they form the pore. Okay, and this is when the pore formation happens, and then the bacteria eventually lose their intercellular molecules, eventually lies, right, or die. But the gram-negative, because of the outer membrane, you have a barrier, right? The bacteriocin cannot get into the cytoplasmic membrane. So if you destabilize outer membrane in the gram-negative organism, and then the bacteriocin can penetrate and can cause damage, so that you can have some limited death or limited inactivation of microorganisms. Is it clear? So is it going to work well in the biofilm setting? It's going to face the same challenge as any antimicrobials, right? Or antibiotics we talked about. The diffusion barrier is there in the biofilm, right? They can be absorbed by the matrix, so may not, and the cells are inactive or dormant, so they may not respond well. So it's the same challenge as bacteriocins as antibiotics. Everybody follow me? The same mechanism or mechanical barrier they are going to face. So it may not work. So again, then you have to break down the matrix with enzymes. That's one strategy. Or you can physically destroy them, right? Then you can drive antimicrobials. Okay. So this is antimicrobial peptide, again depending on how and what level of depth of study you want to do. This one actually published with a collaborator from Purdue. We worked on together, and this student was working on melittin. This is a small peptide, and you can see how they cause the lysis. So she modeled it and how this peptide formed this pore, and you can see that integrate into the lipid bilayer membrane, and then can sit inside, and then you can see how that formed that pore, and that's called water channel formation by this protein peptide. So depending on what type of different protein or peptide, melittin I think from bee honey I think. And then there is another one from the frog skin. That's also another antimicrobial peptide. So because not only bacteria, also mammalian system produce antimicrobials, right? We talked about cathelicidin or tryptine, right? Also defensin. A lot of those are antimicrobial peptide and they work same way. Okay? All right, so they are going to form pore and cause damage. Okay, now let's talk about a little bit about the nanotechnology based approach. Again, I think I'm sure you have learned or read papers, right? Or some of you may be working on nanotechnology based approach. So what our goal is, if you can use nanotechnology or nano carrier, nano device to deliver antimicrobial peptide, right? So for example, nano liposome. We talked about that a little bit. Liposome is a lipid bilayer structure. You can have a multi-layered liposome. You can have a different sizes liposome. What you can do, you can load up antimicrobials within that, and under certain conditions, they're going to deliver this molecule, right? So if you have a biofilm, the nanoparticles, because they can disperse through small spaces, right? That's the advantage. Imagine you have a biofilm, and only the small molecules can diffuse through, penetrate through. So the nanostructure, we're looking at very tiny, right? 10 to the power 6 nanometers, the size, right? And that's the molecule we're going to use to deliver small molecules. Nano emulsion is also another one. You can use a oil droplet, and then you can decorate these molecules, antimicrobial for example, outside to deliver it. Also, this is another one, again, it was worked on at Purdue University with one of my colleagues, and he's a carbohydrate expert, right? So he found some natural dendrimer-like structure. So what he did, he loaded niacin in the core, and as you deliver, whether it's for drug delivery or for antimicrobial purposes for food preservation, you can deliver this, and this model actually allows slow release of this bacteriocin, slow release of antimicrobials. So if you don't have this type of delivery system, what will happen if you just suddenly load it up, like without delivery system, they're going to disappear pretty quickly, right? Because other substance like enzymes, they're going to break it down, or it's going to diffuse everything at one point. But for most applications, you want slow, continuous, controlled release, we call it. So slowly, you'll be diffusing over time so that that antimicrobial effect can last longer. Okay? And that's why the nano scale or nano delivery, nanotechnology approaches are extremely important, are efficient. So that's with the nano subject. But then bacteriophage also can be used, right? Bacteriophage attack the bacteria, and they can attack and they can kill. These are actually, again, a couple of colleagues from Purdue, they isolated these two specific virus. So I got the picture for them, from them for my book. So if you look at this book, I think I used part of these pictures. So typical bacteriophage, you have a head, DNA is encapsulated or packaged, the tail, right? This is the tail, that's the base, and this is how they inject DNA into the host, right? And then the binding is extremely important, right? The binding protein is critical because that's what dictates the specificity. And also the protein they use to lyse the cell, okay? And that's how you destroy it. So we are interested in lytic phase of the bacteriophage action. And these are all different types of bacteriophages, okay? They're not one you saw is a typical one, lambda phase, we call it, T4 lambda phase. It's a typical one, but you can see all different shapes, sizes of bacteriophage. They attack bacteria. So people are studying quite a bit on bacteriophage. So you can find the greatest application of bacteriophage in number of applications, right? So you can do it for treatment. Instead of antibiotics, can you use bacteriophage to treat a disease in humans? Yes, no? It's kind of weird. There are a lot of work going on. So if you follow the history of bacteriophage as a therapy, Russia is the one, I think Eastern Bloc, they started many years ago. In the beginning of this century, they used, not this century, the last century, okay? They've been using this for a long time. There are a lot of old literature. Unfortunately, that literature in Russian language, and most of the people not familiar with the Russian language, but they started using bacteriophage for therapy. Whether it's a diarrhea or other cases, they started using it. So now we are coming back and we use it again for treatment of diseases. And just briefly, you know how they cause lysis, right? So once it's being attacked by the bacteriophage, it's going to release the DNA, right? and what you want actually DNA to integrate. Eventually they will multiply, they will repackage and burst open, kill them, right? But you don't want the lysogenic cycle, because that's not going to kill the bacteria, right? It's going to maintain it. So you need to know the life cycle, and you can control the lytic cycle versus lysogen. Okay, that is a way you can do it. But most of the time, you prefer this to kill the bacteriophage. Okay, what are the factors? How does it work all the time? Is it going to work well in the biofilm? And if so, which cells are going to be killed? Okay, think about all the biofilm layer, right? Multi-layer. So let's start this on the surface layer. So that's where they're more exposed to, right? And they're going to be affected or killed, right? What about the cells which are dormant sitting inside? Less likely, right? They are not functional, they may not take it, they may not divide, so therefore bacteriophage is not going to divide, and then this cycle is not going to be completed, right? So it's going to be fruitless, or it's not going to be fruitful, right? So it will be useless, kind of. So it's going to have a limited application. But then again, you're trying to kill them when they are actively growing, right? Actively dividing, that's when it's going to work. So in the biofilm applications, you might find, well, limited. So now let's see. You know your certain surfaces, there is a biofilm formation, right? And I want to use bacteriophage. How can you use bacteriophage to completely eradicate biofilm? Okay, so we talked about the problems, right? Now I'm giving you the question. I'm going to, as a student in my lab, I'm going to give you this project. Find out a solution for inactivating biofilm or biofilm forming cells using bacteriophage. And what strategy would you use to ensure that all the bacteria in the biofilm, they're going to be killed, so that I can eliminate the the biofilm problem from my industry or food or what, a catheter or medical device. So I already gave you the answer. You already know the answer. Okay, I don't know whether you can explain it, but you already know the answer, right? So you need to make them what? Active. So first step I will do, I will disperse the biofilm, mechanically or enzyme treatment, right? And then what I would do, I will try to make them grow, right? I don't want persistent cells, I don't want dormant cells. If I can change their nutritional condition, right, I want to make them grow, make them feel happy. And when they are happy, then hit it with bacteriophage. You see this? That's when you kill them. Not when they are depressed and sad. Don't try to kill them that time. When they are happy, means the bacteria growing and they are physiologically active, and then that's when you add the bacteriophage, you kill them. Okay, now you eliminated that. Then your problem is that I want to continue to use biofilm for preventing, continue to use bacteriophage for eliminating biofilm. Even for disease condition. So like I want to treat it. Is it going to be useful or effective after certain time, certain period of time? So first time you had bacteria, right? Or you made this happen, so the most of the cells are dead. So can you continue to apply biofilm so that you don't have that disease reoccurrence? He said no. That's a good thought. Anybody else? Just say yes or no. You know how the answer is. So you need the substrate, right? You need the host. So once you eliminate, that's the problem with the bacteriophage. Once your target is gone, the bacteriophage, is it going to be there around? That's the problem, one of those. That means we have to have some level of bacteria always maintained so the bacteriophage can replicate. But if the host is gone, usually they are going to disappear too, so you have to continue to apply, right? So that's one of the challenges with the bacteriophage. Everybody followed that? It's not like antibiotic, right? Antibiotic is going to maintain in the blood circulation for a while, and if there's a pathogens growing, they're going to be killed. So you maintain that dose for a certain period. But bacteriophage, once the host is gone, essentially they are no use. They may disappear as well. Until next time your host is back, host means in this case bacteria, then the bacteriophage is going to work, right? Okay, conceptually make sure you think about that. Okay, bacteriophage application. So I already mentioned therapy, control, right? Prevention, you can do it, but again, that's the limitation you're going to have. You have to have your host bacteria and sanitation. So all of those applications you can apply phage. Desirable properties of bacteriophages for food safety application. Usually it has a broad host range. Strictly lytic, we are looking for lysis of the target. Propagated on a non-pathogenic host. They can propagate on non-pathogenic. So this is another challenge though. So if you are trying to control salmonella, right? So you have to grow bacteriophage in salmonella, right? So that means if you are trying to market that, commercialize it, so we are going to produce large amount of bacteriophage specific for salmonella, you have to grow large volume of salmonella. You see this? That's a nightmare. Because if this accident happens, if you are a bacteriophage producing company, and you are going to kill a lot of people with salmonella, because you have to grow a large number. So somehow, I remember one of my colleagues, he's working on, Bruce Applegate, working on bacteriophage. So always they're finding, can you use a non-pathogenic host, or engineered host, so that they can harvest a lot of bacteriophage. That's a challenge. Like E. coli 15787, which is one of those deadly enteric pathogen. It causes severe foodborne outbreak. And so if we are going to make bacteriophage for E. coli 15787, you have to use E. coli 15787 to grow it. And when you grow large number, and it's the people who are working in that facility, their health could be in danger. Also, if there is a exposure. Or how do you purify the bacteriophage so that you don't have any bacteria in the bacteriophage preparation? Are you following my logic? So that means instead of treating, you may reinforce that infection. That's always a challenge. Keep that in mind. Hopefully, they are going to solve all those issues, but these are actual practical problems. We know the complete genome sequence, non-transduction, non-viral transduction, absence of any genes encoding pathogenicity. You need to have that. Make sure it's not carrying any gene for pathogenicity. Oral feeding studies show no adverse effect. GRAS approval, generally regarded as safe, has been approved, some of them. Sufficiently stable under storage application conditions. So they need to be stable in order to use it. So this table, again, you may not be able to read it. You can, the slides you're going to get it. What is saying actually, what are those bacteriophages now approved or you can commercially buy? Okay, these are some of the company marketing it. These are the product. Again, you find it again, Listeria, E. coli, Salmonella. These are the different bacteriophages they are using. These are the target organism. Okay, and this is the website. If you are interested to find it, or you can Google it, you can find that. So these are some of those commercially available bacteriophages they are trying to market. Okay. Okay. So as I said before, a lot of times the bacteriophage may not work in the deeper into the cells, and I already mentioned you want to disperse them, right? You want to apply enzyme, and that enzyme can break down the matrix, then you can apply it. So here is the example. In this paper, you can read it. Enzyme can break down the matrix, and then it allows the bacteriophage to get in, and then eventually target the bacteriophage and kill it. Okay. The other bacteriocin also same story. I may not be able to reach that. So hopefully enzyme or some of this treatment allow the bacteriocin to reach to the individual cell. That leads us to porous sensing inhibitors. You mentioned that communication stop. Okay, so how do you do that? Review. So the Lux operon going to produce this sensing molecule. Acyl homoserine lactone is one of the major molecule, right? And then it binds back, and then it helps transcription of the gene. and that can lead to biofilm formation, sporulation, becoming pathogenic, they become motile, and also sometimes there will be bioluminescence, right? I think you heard that lecture on that day about Vibrio, and so they can be bioluminescent. So these are some of the functions. So this is the function of AHL. And we also learned that it's a cell density indicator, right? At low cell population, they are not going to make it. At high cell population, they are going to produce the quorum sensing molecule. So this is the way they produce. They bind to the receptor and activate. This is in gram positive. I used the gram negative as an example before, just to review it. And so now, again, this is kind of like telling you the same story. So we are using all those different applications to control the bacteriocin. QS inhibitors, if you look at the list of chemicals, and I know many people working on it, especially the plant-based molecules are getting very popular because these are natural, these are sustainable. Can you use these molecules to stop their communication? They're going to inhibit those quorum sensing molecules. So these are some of those list of chemicals. Some of this coming from grapefruit juice, right? These are acylase that breaks down the AHL, and also a lot of essential oil, thymol, carbachol from grape or eugenol. These are all of those come from spices and herbs we eat. So those also can inhibit the quorum sensing. And these are well established nowadays, and people are trying to use that. Can we use that for biofilm control instead of trying to use antimicrobial agents? These are more natural. If it mixed with food, probably it's not going to be harmful because we eat that. These are grass listed, so that worked very well. So people are trying to find out all different types of natural, grass listed, plant derived molecules as anti-biofilm agents. Okay. These inhibitors again work same way. Somehow, if you can degrade this, right, this QS molecule getting in, and also you can prevent binding. Can you use antibody to prevent binding or neutralize the QS molecule, right? So that's been proposed. Also, some of the speakers mentioned that. And this is the AHL homocysteine. You can clip this part, then this become a non-effective or non-active or unactive or inactive molecule, right? So if you cut it out. So this is how essentially works. And these are other things also. You can combine it with the linkers antibiotics, right? And we know quorum sensing molecule, they're going to bind to the receptor. This is a fascinating way you can control it. We know quorum sensing molecule bind to the receptor before it's taken into the cell. So can you tag antibiotics with quorum sensing molecule? Because they have to bind, and then the antibiotic can get in, and they can destroy the cell. So this is another way you can do it. Okay. I talked about the microbubble, nanobubble. This is kind of graphical presentation. So if you create ultrasonic wave, and the bubble, the water bubble, for example, right, or surfactants like a soapy water, right, it forms these tiny bubbles, and so this bubble can change the shape, and at some point, this smaller one is going to be bigger, right, because the small one coalesce, they call attach together, and then fuse together and become large. And so what happens actually when this bubble bursts is called cavitation, okay, and that's an energy differential, and that can actually disrupt the cellular architecture, including biofilm. And this is recent, relatively new technology. I got this in 2025, but I know some of our colleagues also working on this called micro nano bubble technology. Okay, and this bubble, when you can physically form them, and then there is a time energy differential, you can allow them to burst, and that burst creates certain cavitation. That's what we call it. So it's an energy differential, and that can hit microbes, tiny molecules, in a certain force. They rupture, they break open. Okay, and that's becoming important because it's like a non-chemical treatment, right? But you are just using some physical energy, right? In this case, ultrasound, and you can create this, and then you can get rid of microbial biofilm or microorganisms. And people are trying to use that also food processing. conditions, some applications to kill the microorganisms. I already mentioned some of our work. I will just last two approach we are using to control biofilm. So already I talked to you about tunicamycin, right? How tunicamycin works, antibiotic, and also it a good biofilm buster, right? So at different low doses you can control it. Again, just to review it, this is the lipoteichoic acid attached to the peptidoglycan, and this pathway is regulated by the small molecule called tunicamycin, right? And so you want to prevent that formation of teichoic acid or peptidoglycan. As a result, you can prevent biofilm formation, right? And I showed you data for the structural attributes I mentioned, and also structure in Listeria and structure in Staphylococcus. And the most importantly, the morphology changes, colony architecture changes, and also the biofilm, right? So at different concentration, you can see if you go higher concentration, the biofilm formation become more dispersed, because we know the molecules which are responsible for biofilm formation, you can break it down. And so that means when you are using this molecule, I think a lot of people are trying to use this tunicamycin as a therapy because of the resistance to penicillin, right, which is very common. And so for that, they are working in the cell wall synthesis in a different way, and people are thinking, can we use tunicamycin as a treatment for some of the diseases, okay? Because it clearly shows not only just the biofilm formation, it also affects other properties. We already mentioned that. And so here, you can also prevent their attachment to KCO2 cells, right, at different concentration, higher the concentration, low attachment. And it could be function of you are killing the cells, and also at the same time, they're not producing this peptidoglycan and teichoic acid, okay? That's the way you can control it. So other one we did is the called smart antimicrobial nanoparticle. I already showed you the structure, right? So why do you call it smart? Smart in this sense, I don't know how you use this word here in India or some other countries. Here smart means we are talking about intelligent, okay? Is it an intelligent molecule? It can go and find the target and bind and destroy it, okay? It's almost like a laser guided missile, right? It go and find the target and just specifically kill that. So I will use this, hopefully I can explain it very quickly. So in this case, if you design a chitosan based nanoparticle, chitosan is actually a, the shrimp or crab, there's a large amount of carbohydrate, right? It's kind of, people are finding a lot of use of it. A lot of medical application people are finding. And the chitosan, the way it works is that at large molecular weight, it's not antimicrobial. But at low molecular weight or certain molecular weight, it become antimicrobial. That's the properties that are very fascinating. And so what you can do, or people are doing actually, you can create this chitosan nanoparticle. You know how to synthesize nanoparticle. And then you can tag it with the antimicrobial peptide. Nanoparticle, antimicrobial peptide. So now you can put something, a target finding molecule, right? So think about the last one which talked about quorum sensing molecule, right? You tag it with antibiotic. Can you use something like that that can find the receptor and bind there? So in this case, actually, we used Listeria adhesion protein. I mentioned that. And Listeria adhesion protein binds back to bacteria. Okay, when it's secreted, it binds back. So we said, can we use this LAP molecule on this nanoparticle with the antimicrobial peptide? Can it go and find Listeria and kill it? That's why we call it smart. Okay. So we published it. Actually, it works because that's what I told you before. LAP is synthesized and it comes and binds back. That's the beauty. If it binds back, then if I load up any nanoparticle with this LAP, right, with the antimicrobial peptide, then think about this fate of this bacteria is going to be killed. So we wanted to prove that. So we created this nanoparticle, and then nanoparticle can diffuse through the biofilm because this is a small structure, 100 to 200 nanometer, and that's the beauty of it. And then eventually it can break down the cell. But again, you can have a problem because the dormant cell or persistent cells may not effectively show the result or response. It may be very effective against early biofilm stage, like a 24-hour, 48-hour old biofilm in the beginning, when you have more cells are active and fresh. So you can do that. Other thing we also did also epsilon polyl lysine. And if you heard about polyl lysine, that also antimicrobial. You tag that as an antimicrobial agent. So first thing, I think this is a postdoc, he worked on it, and he created, in this case we use gold nanoparticle. Let me tell you first about the gold, and then we'll talk about chitosan. And so he created gold nanoparticle, and this gold nanoparticle you can see clearly binds to the, with or without bacteriocin, it can bind to the surface of the listeria. You can see coated with that. And so what we wanted to prove is that if you have a bacteriocin loaded gold nanoparticle, and if you look at the death, killing, and essentially it's inactivating all the listeria species. You can see the clear zone. But now we have this one nanoparticle with the lab attached to it, just to see if it can show enhanced activity, because it's going to find the listeria, can bind it and kill it. And so if you look at this, I don't know whether you can see, all of you, and you have a actually increased zone of inhibition, little bit, at least. Ivanovia also has the same function. You see a little bit. So if you look at this statistically, there is a little bit difference or higher killing. So the hypothesis we came up with, that if you create SMART, find a target, it binds and it kills more. So those are the creative way you can use during your research. So, well, I got a system which can kill it, but can I enhance its activity? Because you have to find out they bind to certain receptor on the surface. If they bind it, they can kill more. This is a common sense application. This one nanoparticle, so we are looking at cytotoxicity. If this nanoparticle, if it is accidentally consumed by you, is it going to cause any damage? You can measure this cytotoxicity. I already told you about cell-based assays we did before, right? You can use that to verify are they cytotoxic. So these are also, if it can prevent adhesion of listeria by treating this with the nanoparticle. So can it inactivate biofilms on the plastic? We use that on the plastic biofilm. And then also, this is 24-hour old culture. This one is 48-hour old culture biofilm. And you can clearly see this is 6-log reduction. I mean, it's a huge reduction by this lab or the smart biofilm. You can see this clearly is killing it. This is the gold nanoparticle with the lab, and then the pediocin attached. So this has a good inhibition, but this one has showing the maximum inhibition. So this created the SMART means finding the receptor, binding to it, and is inactivating them. And other application you can do right on the plate. You can do it on the conveyor belt, where actual problem is in the industrial setting. You can use that to inactivate biofilm forming microbes. And these are good ways to monitor it. So if there is a dark color, that means these sample areas are positive. Because remember, the biofilm can form in the places where you cannot use the sanitizers to inactivate microbes. And that's the problem, because they are living in a lot of different places within the food production industry. Okay, and so like I was saying, that they are growing in certain environment, and your disinfectant cannot reach there. And so if it is a conveyor belt, this is a small laboratory scale, but if you think about long conveyor belt, have you seen that? I mean, this conveyor belt is used for transportation of food, right, in the processing plant. If they produce something, they are going over the conveyor belt, they are going to be packaged, and then they deliver, right? This is the whole system. And so a lot of times, all of those places, the microorganisms can be throughout the conveyor belt. They can form biofilm, and they become a continuous source. So the factory, what they do, they disinfect and use disinfectant or sanitizers to kill the biofilm. Okay, and so here we are trying to prove that this can be used as a spray to control it. Last part, I'm going to talk about the chitosan-based nanoparticle. And so the student he synthesized it, he conjugated the polyl lysine, and then you tested if you have a chitosan nanoparticle, just the chitosan nanoparticle conjugated to polyl lysine, which is the antimicrobial. We wanted to see if they inactivate microbes, right? and so you are measuring the minimum inhibitory concentration. So CHNP, chitosan nanoparticle, if it has a 25 to 37 MIC value, and here you can see very low doses, you can achieve the same killing, right? So that means conjugated chitosan nanoparticle with antimicrobials is more effective than the chitosan nanoparticle alone. Okay, this is just a proof. You can verify that. These are non-toxic, so we always measure that. And we are looking at the biofilm adhesion in this case. And so you are using chitosan nanoparticle on the cell line and try to find out if they're going to be toxic to the cells. But the most importantly, can it prevent biofilm formation? Can it inactivate preformed biofilm? So here we use Pseudomonas, Listeria, Staph aureus, Pseudomonas, Salmonella, and E. coli. And you can see this chitosan nanoparticle can significantly inhibit. Except Staph aureus, you get lower inhibition. But in most cases, they can prevent biofilm formation. If we apply it on certain surfaces, they can prevent it, right? We want it. And this, if you look at this, we are looking at violet stained, right? If it is untreated, you expect them to form biofilm, right? But here, when they apply the chitosan nanoparticle, they can inhibit. In this case, could not. That correlate with this data, and here also. But as soon as you have a chitosan nanoparticle, all the blue color gone. That means the biofilms are being eradicated or eliminated. So that's confirming this data. What about preventing mixed culture biofilm? So prevention, or excuse me, this is also preventing mixed culture biofilm, and we saw that it's very effective. Again, it's proved by that whole process. Staph aureus, Listeria, Pseudomonas, Listeria, and we wanted to see if this is effective. So it's clear means they are killing this mixed culture biofilm. So can it inactivate preformed, already biofilm formed? Can they go through the biofilm structure? Can inactivate them? And that's why we saw less effectiveness. And so there are some decrease, but some cases there is no result or no activity. So that's the bad news, right? Mixed culture inhibition is low, but you still get some inhibition. So that's one way you can inactivate it. I think this is, I want to just quickly go over that. This is for the seed. This is alginate-based antimicrobial coating reduces pathogen alpha-alpha on the seeds. So this is the seed architecture where the bacteria would be, and they use a lot of this technology to eliminate Listeria and Salmonella from the seed surface. You saw that on the cantaloupe surface. One of the clever things they did actually called brushing and removing this biofilm. So physically you can break it down. So what they did, this is our colleagues, they did that. They had a brush set up on this biofilm, and then they had some antimicrobials. And so what you do actually, you rub that surface, and then you try this application, and then if it removes it. So they were effective in always the biofilm. If you have it, you have to physically break it down and then apply your antimicrobials, and that can effectively remove the biofilm. So their studies showed that you can effectively remove this biofilm structure. So these are some of those fascinating technology or approaches that can help remove these biofilms on the produced surfaces. Okay. Clinical approach, quickly, I didn't realize that I have the clinical as well. You want to take a break now? I know you guys have time for break anyway. Okay, let's take a break, say half an hour or so. I think you're supposed to have your coffee break or tea break, right? Let's have that. Then we can come back and I'll finish it. The idea is that we can finish it before lunch. But today you're going to have a little late lunch because I got the other chapters to finish. Okay? Okay, let's do that. Take a break, and then I'll finish the last part. This is